Well, hello, future nurses. Let's talk about genetics. Genetics is the study of inheritance. It began formally with the work of Gregor Mendel in the 19th century. He was an Augustinian monk working in the modern-day Czech Republic, and his work was extended by many people after that. But it really came to a head with the discovery of DNA by Watson and Crick and Rosalind Franklin. As the slide says, this is the molecule of inheritance, deoxyribonucleic acid, or DNA. Let's take a look at this molecule in some detail. So we have a sugar phosphate backbone, like this. In fact, we have two sugar phosphate backbones. This is a double-stranded molecule. The sugar is a 5-carbon sugar called 2-deoxyribose. The phosphate is charged. The sugar is very, very water soluble. So this backbone really likes the water. Inside this double helix structure are the nitrogenous bases, and there are 4 of them. T, which stands for thymine, A, which stands for adenine, G, which stands for guanine, and C, which stands for cytosine. In DNA, thymine always pairs with adenine, and adenine always pairs with thymine. Cytosine always pairs with guanine, and guanine always pairs with cytosine. We call this type of nitrogenous base pairing complementarity. I'll just say complementary. Complementary. C-O-M-P-L-E-M-E-N-T-A-R-Y. Complementary means if you have a thymine on one side, you have to have an adenine across from it. If you have a guanine on the other side, you must have a cytosine across from it. If you have an adenine on one part, you must have a thymine across from it, and so on and so forth. Now, these bases don't like water. In order for the molecule to reduce its exposure to the water, the molecule twists upon itself. This is called a double helix arrangement. The other interesting thing about the double helix is that this part of the strand over here, that's the front right here. That means over here, this strand here, this is the rear of the molecule. And then if you follow this strand down here, this molecule down here, well, that's the rear of this molecule. But this other molecule over here that we trace to over here, that's the front. So these two strands are in opposite orientations. We call that antiparallel. These two strands must be antiparallel. If they're not antiparallel, they don't fit together. This little cartoon, I think, shows you the structures of the various components of DNA quite nicely. You can see the 2-deoxyribose is a 5-carbon sugar. So the carbons are 1, 2, 3, 4, 5. And in order to distinguish the carbons in the deoxyribose from the carbons in the bases, the carbons in the sugar are given a little prime after them. So you have 5 prime, 4 prime, 3 prime, 2 prime, 1 prime. Those are the sugars of the 2-deoxyribose. The phosphates, as you can see, are here and here. Phosphates are attached to the 5' sugar, 5' carbon of the sugar, 3' carbon, 5', 3', 5', 3', 5', 3', 5', 3', so on and so forth. This strand is going from front to back. The front of the molecule is called the 5' end. So front and 5' both begin with F. The 3' tail of the molecule is the back end. And then this one goes from front to back. That goes from back to front. That's your anti-parallel arrangement. And that one down there, my picture is there, so you can't really see that. Here are your bases. They are complementary. Notice that they bind to each other via these hydrogen bonds like this. These bases, again, they don't like the water. Notice some of these bases have 2 rings, like adenine and guanine. Some of these bases have 1 ring. Bases with 2 rings are called purines. And these bases with just 1 ring are called pyrimidines. Cytosine and thymine are the pyrimidines. Oh dear, that's an R. Pyrimidines. And if you want to simplify the structure of DNA, here's your backbone. Sugar phosphate, sugar phosphate, sugar phosphate in an anti-parallel arrangement. And here are your nitrogenous bases. We'll just say B for bases. And this will twist about itself. Now this molecule is very regular. You saw that in the last slide, so that every turn of the helix from here to here is 10 bases. There's 10 bases per turn of the helix. This molecule is very regular, so that you have a particular width to the molecule, and 10 bases gives you 34 angstroms. From one turn to the other. That means every base, there's 3.4 angstroms between each base. And the molecule is about 20 angstroms in width. Very regular molecule. Now, how does this molecule store genetic information? The answer is in the sequence of the bases. And that's shown in the next slide. The sequence of the bases are read in 3s. These 3s are called a codon. And each codon codes for a particular amino acid. And that particular amino acid will then be strung together to form proteins. Some of these codons don't code for anything. They are stop codons. So that tells you where to stop putting amino acids in. This amino acid, methionine, is used to start the process of protein synthesis. So DNA codes for protein by having a sequence of bases that is read in 3s. And those 3 base motifs are called codons. And these different sequence motifs code for different amino acids. DNA is converted into RNA. I think a more accurate way to say that would be an RNA molecule is made using the DNA sequence as its pattern, as its template. That process by which you make RNA, an RNA copy of one of the strands of DNA, is called transcription. Transcription in eukaryotic cells takes place in the nucleus. The enzyme responsible for transcription is RNA polymerase. There are 3 RNA polymerases in eukaryotic cells. RNA polymerase 1. They're designated usually with Roman numerals. RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1 makes ribosomal RNAs. So it transcribes all the ribosomal RNA genes. RNA polymerase 2 will make your messenger RNAs that code for proteins. RNAs. Oh, that's messy. Yep, you get the point. And RNA polymerase 3 makes tRNAs. Also makes a small ribosomal protein called the 5S ribosomal RNA. Then it makes some small RNAs that are used for regulation. These RNA polymerases begin at a sequence of DNA. They engage the DNA at a sequence called the promoter. The promoter is bound by proteins called transcription factors, which we're just going to abbreviate TFs. When those transcription factors assemble on the promoter, the RNA polymerase can bind to the promoter and initiate transcription. After it clears the promoter, it will start at the 5' end of the RNA, and it transcribes until it gets to the end, and then it will disengage from the promoter. And I drew the promoter here over here, and that is incorrect. The promoter would have been over here, since it would have started over here and would have gone this way. I missed the arrow there, sorry. These 3 different RNA polymerases utilize distinct promoters. They're distinct sets of promoters, distinct sets of transcription factors. There are a few transcription factors that are held in common between these 3, but for the most part, the transcription factors differ for each of these RNA polymerases, and the promoters have very different structures as well. Those RNAs are taken to the cytoplasm from the nucleus. They're usually highly processed as well, and they're given to a RNA protein complex called the ribosome. And the ribosome will read the codons in the sequence of the messenger RNA to make protein. We call that process translation. Translation, there is an initiation step to translation. If I use yellow, you can probably see that better against the orange here. So initiation of translation begins with the methionine tRNA, and it's charged with a methionine amino acid. You then have proteins called initiation factors, which are abbreviated EIF, and then they have a number usually, some sort of number. You need the particular initiation factors, the initiator tRNA, that's with the methionine attached. You need those assembled on the ribosome, and then you can initiate protein synthesis. Protein synthesis is shown in the next slide. This is called the elongation step. As the ribosome moves to each of these different codons, different tRNAs with the proper RNA move in, they will attach to the ribosome. So here's your AUG, there's a P site, and then the incoming charged tRNA attaches to the A site. That occupation of both these A and P sites with charged tRNAs initiates the large ribosomal subunit to form peptide bond. So the peptidyl transferase is actually in the RNA of the large ribosomal subunit. It is a ribozyme. When the peptide bond is formed, another protein binds, a translocase binds, and causes the ribosome to move in the 3' direction down the tRNA so that you now have new codons at these sites. The A site is open. You have a new charged, the appropriate charged tRNA comes in that corresponds with this, whose anticodon sequence corresponds with this codon sequence. And the process repeats. The large ribosomal subunit remakes the peptide bond, and it moves again with the translocase, and away you go. When the ribosome gets to one of those stop codons, so the stop codons are UAA, UGA, UAG, there are no tRNAs that recognize those stop codons. Instead, the ribosome will pause and proteins called release factors will bind to the stop codon. The release factor will cause the protein, the nascent protein, and the RNA to dissociate from the ribosome. The large and small ribosomal subunit will then fall apart, and translation has ceased. DNA replication is a very complicated process. You must unwind the 2 strands of DNA first. The protein responsible for that is called the helicase, that you can see here, the helicase. This is an ATP demanding process. You then have DNA polymerases that can make DNA. They can't make it at the first, they have to have a primer. That primer is a strand of RNA that allows them to add new nucleotides to the 3' end. So synthesis goes from the 5' to the 3' end. All polymerases go from 5' to 3'. This one, as you can see, can start at the end and go straight this way and can work as fast as a helicase can unwind DNA. These free nucleotides are actually deoxyribonucleotides. So deoxy ATP, deoxy TTP, deoxy CTP, deoxy GTP. The polymerase will use the energy in these triphosphodeoxyribonucleotides to form these phosphodiesterase bonds in this strand of DNA. The RNA primer that is made so this polymerase, DNA polymerase, can get a start is called a primase. And the helicase keeps unwinding on the lagging strand here. This has to continually start and stop. And so on and so forth. So you'll have bits of RNA in here, and there are repair enzymes that come in, take that RNA out, replace that DNA, and then there's an enzyme called DNA ligase that seals the gaps left in the backbone. So DNA replication is very complicated. It takes a lot of enzymes. Fortunately, it's a pretty accurate process. Makes a mistake about 1 every billion bases. And then those mistakes can then be fixed by DNA repair machineries that are available to the cell as well. This shows you the processing of a messenger RNA that are made by RNA polymerase II, the front 5' end has a cap put on it. The 3' end, when transcription terminates, has a polyadenine tail put on the 3' end by a polyadenyl polymerase, a PAP. This tail is about 100 to 250 adenines long at the 3' end. So this process here is called capping. Capping occurs actually very early after the initiation of transcription. Tailing occurs in combination, essentially coupled with transcriptional termination. Then there are pieces of the transcript that are removed. These pieces that are removed are called introns. This removal of introns is called RNA splicing. RNA splicing is executed by an RNA protein machinery called the spliceosome. The spliceosome. RNA splicing allows you to make multiple protein coding regions from the same coding region. By making one nascent RNA, you can splice it differentially and get lots of potential proteins out of it. That way the cell can make a lot from a little bit of the genome. Very efficient use of the genome. And this shows you essentially that process, capping at the 5' end, tailing at the 3' end, and introns through RNA splicing. RNA processing also occurs in the nucleus. Changes in DNA can occur. The polymerase can make a mistake. It doesn't make a mistake very often, but it can make one. And sometimes mistakes in DNA can be missed by the repair machinery. If the repair machinery is overwhelmed, then it will miss mistakes. Mistakes in DNA may change the amino acid that is inserted during translation. You can see here, this sequence is UUC, UAA, GCA, GCU. If you have a change from GCA to GGA, so here we have UUU, UAU, GGA, GCA. So the GCU is still arginine. So that one's fine. The glycine, the alanine GCA is now GGA, and that's glycine. So that mutation changes the amino acid that's inserted. UAU, that doesn't change. That's still a tyrosine. Now UUC is changed to UUU. That doesn't change the amino acid, however, that you put into that position during translation. That is called a silent mutation. You change the sequence, but you don't change the amino acid that is inserted during translation. Now this mutation here, however, that changes the amino acid sequence. So this is called a missense mutation. Some proteins are very sensitive to missense mutations, and one missense mutation in the right place can modify the function of the protein. They can diminish the function of the protein. They can completely wipe out the function of the protein, Or in some cases, they may not change the function of the protein at all. Depends on the protein. Now here, if we change the UAU to UAA, so we've changed this amino acid right here. Now we convert that into a stop codon. And during translation, you'll end up with a truncated protein. That's called a nonsense mutation. Nonsense mutations usually produce protein that is non-functional. And these are usually pretty severe. Likewise, you can add or you can delete groups of bases that are not multiples of 3. You'll notice that the cell reads the DNA in groups of 3. If you insert or take out 3 bases, you'll take out 1 amino acid, you'll add 1 amino acid. That may not change the protein. However, if you add multiples of bases or take out multiples of bases that are not multiples of 3, you add 1 base, you add 2 bases, you add 4 bases, you add 7 bases, you add 5 bases, you take out 2 bases, you take out 4 bases, you take out 5 bases, 7 bases, so on and so forth. Now you change the reading frame. So this reading frame here that went phenylalanine, tyrosine, alanine, and arginine, now you change the reading frame and you go isoleucine, cysteine, threonine. You completely change the amino acid sequence. These are called frame shift mutations. You have frame shift insertions or frame shift deletions. These also change the sequence of the protein after the site of the mutation. These will tend to produce proteins with no function. Alternatively, this is rare, but it does happen, you can get proteins with completely new functions. So these mutations can change essentially the coding capacity of the genome. So this illustrates what I was talking about. Silent mutations that don't change amino acid that you insert into the DNA. These typically are in the 3rd position of the codon. Nonsense mutations that generate stop codons. Missense mutations that change the type of amino acid you insert. And this type of missense mutation changes it from one basic amino acid to another. If you don't change the type of amino acid, that's called a conservative missense mutation. If you completely change the nature of the amino acid you've inserted, so you have a basic amino acid here, now you have a polar amino acid you've inserted, that's called a non-conservative missense mutation. These will tend to alter protein function much more than these. You can also take out or add whole bits of chromosomes. These are called gross chromosomal mutations. These usually require breaks in chromosomes. These tend to be caused by things like ionizing radiation, high-level ultraviolet radiation. Certain chemicals can cause breaks in DNA. There's an entire group of anti-cancer drugs that are used largely for leukemias and lymphomas called anthracyclines. Bleomycin is an anthracycline. It's also used for testicular cancer and other types of cancers. Those can cause breaks in DNA as well. Reiterations of portions of chromosomes, those are called duplications. If you have 2 breaks at a chromosome and then this heals, but the sequence of the chromosome gets inverted, that's called an inversion. If a piece of a chromosome goes missing, that's a deletion. If a piece of DNA from someplace else gets put into a chromosome, that's called an insertion. And then if you swap bits between non-homologous chromosomes, those are called translocations. Alternatively, you can have changes in numbers of chromosomes. These occur because of errors during segregation of chromosomes, typically during oogenesis or during spermatogenesis in some cases. So errors during meiosis can prevent chromosomes from properly separating. During meiosis 1, that will tend to cause autopolyploidy. Plants tend to tolerate autopolyploidy pretty well. Animals don't. Humans really don't. So polyploidy is a change in chromosome number. The alterations we showed you in the last slide, those are alterations in chromosome structure. Chromosome structure. Instead of having whole sets of chromosomes duplicated, you can also have single chromosomes duplicated or single chromosomes taken away. And these, again, are due to errors that occur during meiosis. So these will leave you with one extra chromosome or one missing chromosome. These are called aneuploidies. And these tend to not be compatible with life. The only one that's compatible with life is trisomy 21. There are a few others. Some kids will survive with them, but they don't survive usually all that long. Klinefelter syndrome is due to aneuploidy of the sex chromosomes. So a person with Klinefelter has 2 X chromosomes and 1 Y chromosome. And they have these particular feminizing features. They're phenotypically male because they have the Y chromosome, but they've got that X chromosome, and that slightly feminizes their bodies. You have gynecomastia, reduced body hair. You've added fat at the hips. You have some osteoporosis as well. Reduced facial hair. These individuals tend to be tall and lanky. They also tend to have hypogonadism. Turner syndrome is found in individuals with a single X chromosome. You can see these individuals have webbing at the neck. They tend to have short stature. They tend to have hypogonadism. It was thought at one time that there were some mental challenges, mental deficiency. That turns out not to be the case. In fact, I had a student who had Turner syndrome, and she's an attorney now. So people with Turner syndrome, however, are typically either subfertile or not fertile at all. Here's a gentleman with Down syndrome, and you can see the characteristic elongation of the epicanthical folds. You can also see usually that this part of the jaw tends to be kind of large and bulky. Also notice that the neck tends to be short, and there usually are intellectual challenges with Down syndrome kids, but that varies wildly. Now, I have a friend who has a Down syndrome child, for example. He's a computer programmer. So this varies, and it varies pretty wildly. Down syndrome is due to trisomy for chromosome 21. Some Down syndrome individuals will have extra bits of chromosome 21, not a whole chromosome 21. Other types of aneuploidies include Fragile X syndrome. Chromosomal breaks can result from anthracycline drugs, like, for example, bleomycin. This shows you essentially certain types of breaks and things like that. This shows you essentially chromosomes where you have translocations. So the breakage of chromosomes produces broken chromosome ends. Those broken chromosome ends are highly recombinogenic. There's a repair system inside cells called non-homologous end joining. They will just join whatever ends they can find together, and those will cause translocations. And this shows you essentially the result of those when you try to pair them during meiosis. Sister chromatid exchange can essentially cause recombination events that give you very oddball chromosomes that are usually not compatible with life or fertility. So genes are inherited as units. That was Gregor Mendel's big contribution to genetics. We call an area on a chromosome where a gene is found, we call that a locus. Different forms of various genes are called alleles. To give you an example, let's take paper and pretend that paper is a gene. There's white, red, brown paper. All of these are still paper, but they're different forms of paper, different types of paper. So they're different alleles of paper, to show you what the term alleles mean. Some alleles, if you get them in different pairing, they're so-called heterozygous. Let's take big A and little a. On two different chromosomes, you have one chromosome here, you have this one gene here, and the allele here is the big A gene. Here's the other chromosome, you have this gene, same gene, homologous gene over here, but its allele is a little a gene. This particular trait is the one that will manifest because this allele is dominant over that one. It's dominant because in the heterozygous condition, that's the trait that manifests. This trait will only manifest in the homozygote, where the two alleles are the same. So some terminology here, homozygous meaning you have two alleles at the same locus, heterozygous, two different alleles at that same locus. The arrangement where you have two chromosomes, two homologous chromosomes, that's called diploid. And humans, under normal circumstances, are diploid for all genes. This has application to medicine. So phenylketonuria, for example, is a genetic disease. It's due to loss of function mutations in the phenylaldehyde hydroxylase gene. Loss of function mutations in the phenylalanine hydroxylase gene, when homozygous, prevents people from properly disposing of phenylalanine. Phenylalanine then builds up in the blood, and phenylalanine can cross the blood-brain barrier, and it inhibits enzymes involved in the formation of neurotransmitters, thereby causing intellectual disability and other types of conditions. So here's the straight skinny on phenylketonuria, PKU, phenylketonuria. This is an autosomal recessive trait. It can cause mental retardation, as you can see, and convulsions, behavioral problems, skin rashes, and a musty body odor due to phenylalanine breakdown products that are released in the sweat and broken down by bacteria in the skin. You, however, can head off these symptoms by putting the baby on a strict non-phenylalanine containing diet very early. And if you do, this child can live a relatively normal life, but they have to be on this very restrictive diet throughout their life. Consequently, Phenylketonuria or hypophenylalanemia is tested, one of the things that babies are tested for. And it's a requirement in most states for babies to be tested for PKU very early on. And if it comes up positive, then parents will be instructed to put the baby on a very strict no phenylalanine diet or very low phenylalanine diet. Now, if you wanted to trace phenylketonuria in your family history, you would make a pedigree. There are certain conventions for making a pedigree. So a circle is a female, square is a male. By drawing a line in between circle and square, that means you have a mating. And then the lines below that mating line show your offspring. If you have diagonal lines like this, that means you have twins, and they're dizygotic twins. If you have this sort of arrangement, that means you have monozygotic twins. So these are identical twins. These are fraternal twins. So my brother and I would be drawn like this. We are dizygotic twins. And if you have records and you're not sure what the sex of the sibling is, you take the circle and you put it on its side, a rhombus like this, a diamond like this. That means sex unspecified. If you know the birth order of the kids, you put the number in the square and circle like this. Now, if the individuals don't have the trait, you leave them unfilled in. If they have the trait, you fill them in. If you know the heterozygous for that trait, you half fill them in. And if that particular trait is known to map to the X chromosomes, you would designate it like this. A slash across either a circle or a square means that that individual is dead. And if this sibling died in a miscarriage, you would make the square of the circle very small. Now, if you don't know the sex, you can simply fill it in very small like that. And proband refers to someone whose genotype, whose genes, whose configuration of genes, genotype, is something you know very well. Therefore, they are a proband. And tests you do refer to that person whose genotype is known, and that's a 50-cent word. Genotype is what some of these genes look like. Phenotype refers to what someone looks like. Phenotype. Phenotype's what someone looks like. Genotype's what their genes look like. So here's an example. There's mom, there's dad, here's their kid, here are their kids. So that means first generation, that means second generation. This son was born first, second son, third son. This mom has the trait. This son has the trait. This is the one you're testing. If you have two lines, that means you have a consanguineous mating. That's mating between siblings or first cousins. So here's a pedigree, and this mom is affected right here. Notice none of the kids are affected. No one's affected here. Only when you have these two matings does someone come up here. That strongly suggests you have a recessive trait. So dad is probably like this. Mom's probably like this or something like that. Or dad maybe was probably, better yet, dad's probably like this. So that means mom would have to be like this. So all these kids are heterozygous and they're unaffected. And over here, you have, well, maybe you have something like this. And so everybody here is probably maybe something like this. And then this one here is like something like this. And so 1/4 of these individuals should be affected. And that's kind of what you see. So this person is this, and these others are either this or could be big A or little a. This shows you a recessive, autosomal recessive mode of inheritance. Where you have it in one generation, it disappears, and it appears in the next one. That's pretty indicative of a recessive trait. X-linked inheritance means you're going to see it typically mostly in males or almost exclusively in males. X-linked traits means it is a sex-linked trait, a sex-linked trait. That's what sex-linked traits mean. Things like hemophilia or Duchenne muscular dystrophy or chronic myelogenous syndrome or color blindness or other types of traits. Those are X-linked traits. Glucose 6-phosphate dehydrogenase deficiency, that's sex-linked as well. Sex-influenced traits are traits that are not necessarily on the sex chromosomes. In fact, they're usually not. But the phenotype is influenced by the hormones produced by that sex, and the good example is pattern baldness. So Dr. Wyman, for example, his hair fell out when he was 20-something. Women, however, can have pattern baldness, but their hair usually falls out in their 50s or 60s. So the phenotype is influenced by the hormones produced by that sex. Sex-limited means that you're not going to have that trait unless you have the anatomy associated with that trait. So traits for uterine defects. Turns out the structure of the uterus is inherited. Are men going to be affected by those traits? No. Testicular defects in males. Are those going to affect females? No, because females don't have testes. And so on and so forth. Some traits are affected by just one gene. Most traits, however, are affected by multiple genes. So those traits affected by multiple genes show multifactorial inheritance, where we have the inheritance of many different genetic loci. Consequently, the inheritance of these traits, or of these various genetic diseases for that matter, are subject to many different modifier loci. So the severity of various genetic diseases can vary pretty wildly from person to person because of this multifactorial inheritance.